the importance of the repair of these factors in the maintenance of germination performance. Now, my work has established important roles for the repair of DNA damage in germination and seed quality, and I'm going to talk about that work today. Now, the seed stage of the plant life cycle is associated with high levels of stress to the genome. Whilst on the mother plant, the developing seed is exposed to environmental stresses such as ultraviolet radiation. However, desiccation and rehydration themselves are associated with high levels of reactive oxygen species and extreme stress to the genome. And in the dry quiescent state, damage accumulates in the absence of repair mechanisms. And this accumulation of damage is exacerbated under adverse environmental and storage conditions, such as high humidity and temperatures. And work in the 1970s and 80s established that DNA repair occurs in the earliest stages of germination. Now, of all the biological macromolecules, DNA is irreplaceable. It is both the template for gene expression and DNA replication, essential for cell division, but it is also the genetic material of inheritance. However, DNA is inherently unstable in the cellular environment. It is continuously under assault from environmental factors such as background radiation, air and soil pollutants, and an immense amount of damage is also sustained from the reactive byproducts of metabolism, in particular reactive oxygen species. And to put this into context, every cell in your bodies will accumulate 6,000 damage products during the next hour. If unrepaired, this damage results in delayed growth, mutagenesis, and cell death. Consequently, all organisms need powerful repair pathways. Now, the take-home message from this slide is that several forms of DNA damage can be accumulated by the double helix structure, ranging from oxidative-based damage through to single and double strand breaks. Now, each of these repair products tends to be repaired by specific repair pathways, and of all DNA repair products, the most lethal is the double strand break. This represents a complete severance of a chromosome and represents cellular catastrophe. Now, a few years ago, we established that repair of double-strand breaks is important to seed vigor and viability. Using Arabidopsis mutants deficient in double-strand break repair, we identified that these seeds are sensitive to accelerated aging. Specifically in this slide, we are looking at the sensitivity of mutants deficient in DNA ligases 4 and 6. Now, these enzymes play important roles in repair of double-strand breaks by catalyzing the final step in which the broken ends are joined together. So here we can see that after accelerated aging and germination for 8 days, that germination of DNA ligase 4 seed, DNA 6 deficient seed, and the double mutant are reduced in comparison to wild type. Now, this work established that double-strand breaks are important determinants of seed vigor and viability. As we have seen, there are other forms of DNA damage. So do these play a role in the maintenance of germination potential? Well, to summarize recent work, we've seen that there are a range of DNA damage products and that each of these is repaired by specific pathways. We have isolated mutants in key factors in each of the principal repair pathways shown here and looked at their sensitivity to accelerated aging. We have identified that all these pathways are important determinants of seed vigor and viability. That is, their germination is sensitive to accelerated aging. But in increasing order of importance from left to right, we have identified that pathways for the repair of damage to a single strand are less important than the pathways which repair the double strand breaks. Therefore, the most severe lesions double-strand breaks are those that are most limiting for germination. So, double-strand breaks limit seed vigor and viability. So how can the presence of a chromosomal break influence germination? We hypothesized that seed vigor and viability could be regulated by DNA damage signaling. So how do cells signal DNA damage? Well, the cellular response to DNA damage is orchestrated by two sensor kinases, ATM and ATR. And in response to sensing of DNA damage, these kinases activate signaling cascades of phosphorylation with hundreds of downstream targets. ATM responds principally to double-strand breaks, whilst ATR responds to replication defects. Downstream responses include the activation of DNA repair proteins, the activation of cell cycle checkpoints. These act to slow progression of the cell cycle to give the cell more time to repair damage to avoid the conversion of DNA damaged products into permanent mutations in the genome. And finally, in the presence of excess damage, we see the activation of pathways to program cell death. So initially, we looked for a role for ATM and ATR in germination through analyses of Arabidopsis mutants deficient in these factors. So again, using accelerated aging, on the left, in the high-quality seed, we see little difference in germination of wild-type and mutant seeds. However, after 7 days aging here, we see the induction of the lag phase in the wild-type and ATR mutant lines, but the ATM mutant seeds, designated by the red line, show significant reduction in the lag phase to germination. And as deterioration proceeds, here after 14 days, we see 20% viability of the wild-type seed lot, but we see that ATM and ATR retain greater viability at around 60 and 80% respectively. So this work indicated that ATM is regulating progression of germination and is imposing, at least in part, the lag phase we see to germination in low vigor seed. We also conclude that both ATM and ATR are influencing seed viability. So in these mutant seeds, we predict that they're germinating in the presence of high levels of DNA damage. So to investigate this on the right, we looked for levels of abnormal chromosomes. Now in the panel on the right, we see an anaphase bridge. This represents a broken chromosome, an extreme form of DNA damage which has been misrepaired by the host cell's repair machinery. Now, in the next series of slides, I'm going to be using the aging regime I've just shown you for the ATM and ATR mutant lines. When we looked at levels of chromosomal aberrations in unaged seeds, we see that both wild-type and the mutant seeds carry around 1 to 2% of their cells showing these chromosomal abnormalities, which is higher than we see at other levels of the plant life cycle. However, in seeds which have been aged, so they've lost vigor but not viability, we see that around 4% of the wild-type seeds are germinating with chromosomal abnormalities. This indicates two things: that the deterioration process is associated with exposure to high levels of genome stress, and that the seed can tolerate levels of DNA damage which other living cells are unable to tolerate. Furthermore, in the ATM mutant seeds lacking ATM, 8% of their cells are carrying abnormal chromosomes. And this leads us to hypothesize that ATM is regulating the progression of germination in order to safeguard the future seedling from damage sustained in the dry quiescent state, which might impact on seedling development and mutations in the genome. And this is supported by our analyses of the seedlings germinated from these aged mutant seed. So in the right, we're looking at the development of seedlings germinated from aged ATM and ATR seeds. These seedlings show reduced survival on soil and delayed development of true leaves, in contrast to their surviving wild-type counterparts. Now, the high levels of DNA damage we see in deteriorated seeds lead us to ask the question, does this lead to increased mutation frequencies in the germinated seedlings? So we can look at mutation frequencies by using lines of transgenic Arabidopsis in which mutations can be observed as blue spots or specks or sectors as shown here. So by counting these spots, we can look at the level of mutations. So when we look at the seedlings germinated from 8 seeds, we identify that in the seedlings germinated from seeds which have lost vigor, as shown before, but not viability, we're seeing around a 3-fold increase in mutations, indicating that mutation frequencies substantially increase upon seed aging. Now, in the next part of my talk, I'm going to talk about how ATM is regulating germination. So one of the downstream responses of ATM in response to DNA damage is the induction of the transcriptional response to DNA damage. So ATM induces a subset of hundreds of genes which is greatly enriched in DNA repair and DNA metabolism transcripts. And normally we only see this transcriptional response in response to non-physiological stresses in plants, such as plants that have been treated with gamma irradiation. However, we identified that, in fact, the transcriptional response to DNA damage occurs very early in germination of Arabidopsis seeds. So here, we're looking at the expression of the marker genes RAD51 and POP2, genes with roles in double-strand break repair. And even in germination of high-quality Arabidopsis seeds, we see a striking induction of these genes is occurring by 3 hours into germination. So as soon as the seed is becoming hydrated, ATM sensing is inducing a high level of this transcriptional response, indicating exposure to high levels of genome stress. And the magnitude of this response, in fact, is equivalent to that which would kill a human being. Do we see this response in other species? Yes, we identified it in a range of plants. Here, RAD51 expression in the germinating barley grain also indicates that we're seeing the transcriptional response during barley grain germination. However, when we take barley grain, which are physiologically capable of germination, but which have not undergone desiccation and quiescence, the induction of RAD51 expression is negligible, indicating that induction of the transcriptional DNA damage response in germination is not a programmed part of germination, but a direct response to high levels of genome stress experienced during desiccation, quiescence, and rehydration. Now, a second downstream response to DNA damage is activation of cell cycle checkpoints. ATM triggers their activation, and this mechanism slows cell cycle progression, giving the cell more time to repair damage, to minimize the conversion of damage into permanent mutations. So what happens in germination? Well, in the germinating Arabidopsis seed, most cells spend most of germination in the resting or G1 phase of the cell cycle. And cells in the root tip only begin to move into the first stage of the cell cycle towards the end of germination. And the first phase of the cell cycle is DNA replication, or S phase. And we can visualize this nicely looking at the fluorescence labeling of newly synthesized DNA. So here we can see the appearance of these green fluorescent DNA as newly synthesized DNA is labeled. And we can first see these, what we call S-phase nuclei. They first appear coincident with the cracking of the seed coat during Arabidopsis seed germination. The pink dots here are controlled Dapistane nuclei. And we can see that the number of nuclei in S phase increases in the root tip as the seeds progress towards the completion of germination. Now we can look at the appearance of these green S phase nuclei to look at the timing of cell cycle activation in our wild type and mutant lines. So this slide is just a reminder of the germination performance of the seed lots we're working with during this series of experiments. Again, we see a little difference in germination in unaged wild type and mutant seeds, but remember that after 7 days aging, we see a lag phase induced germination in the ATR mutant seed and the wild type, but that this lag phase to germination is much reduced in the ATM mutant line, shown by the red line. So in the next slide, we're looking at the timing of cell cycle activation by counting the numbers of green nuclei in S phase. So when we look at the timing of cell cycle activation in unaged seeds, we see little difference, as shown by this line, between wild type and mutant seed. But when we look at the levels of cells entering S phase in the aged seeds, as shown by the red line here, in cells in the root tips of ATM mutants enter S phase, that is, they activate the cell cycle, markedly earlier than their ATR or wild type counterparts. So this indicates that the lag phase to germination we see in low vigor seed is at least in part induced by ATM acting through control of the cell cycle. Now, in the last part of my talk, I want to talk about some very recent work in which we've been looking at the link between DNA damage and programmed cell death in seeds. Now, as we've seen before, one of the downstream responses to DNA damage mediated by ATM and ATR is the activation of pathways to programmed cell death. Now, in plants, the SOG1 is a transcription factor, which stands for suppressor of gamma-1, and this transcription factor integrates signaling from ATM and ATR and controls cell death in the plant response to DNA damage. So by analyzing SOG1 deficient mutants, this provides a powerful way to look at the link between DNA damage and programmed cell death in germinating seeds. So in this work, we first of all isolated new SOG1 mutants using genome editing. So what's their germination phenotype? So it's very similar to ATM and ATR. Again, we see little difference between the wild type and mutant SOG lines, but as accelerated aging progresses, if we look in the final panel after 14 days, we see that the wild type seed loss is showing around a 10% viability, whilst we're seeing around 40% viability in the two SOG1 mutant lines. So we're particularly interested in analysing the roles of these mutants in cell death in seeds. Now we can analyse programmed cell death by using propidium iodide staining. So shown here, using propidium iodide, we can stain tissues and dead cells appear as in red. So this is a control experiment using Arabidopsis seedling roots. On the left, in the absence of DNA damage in both wild type and SOG mutant lines, we see little evidence of cell death. However, in the presence of DNA damaging agents, we can see that significant cell death is induced in the wild type roots, but not in the SOG1 lines. And therefore, SOG1 is acting to selectively eliminate cells with damaged genomes in the actively dividing cells of the root tips. And this provides a highly efficient mechanism of eliminating damaged cells without compromising plant growth. So what happens during germination? So in this slide, on top we see the germination performance of the seed lots analysed. On the left, we're looking at unaged seeds. Again, in the embryonic root, we see little evidence of programmed cell death, either the wild type or the SOG1 mutant lines. However, when we look at deteriorated seeds, here in this seed lot, we're seeing around 60% viability of the wild type, whilst the SOG1 mutant lines are showing around 8% viability. And we identified at a frequency of around 8% of the embryonic roots analysed, we see significant evidence of programmed cell death in the wild type roots, but we never see this in the SOG1 mutant roots. So this indicates that in response to naturally occurring DNA damage, which occurs during seed deterioration, that SOG1 and the DNA damage response are selectively eliminating damaged cells, therefore acting as a quality control mechanism. And finally, as I previously showed for the ATM and ATR mutant seeds, the seedlings germinated from the SOG1 mutant lines show poorer survival on soil than their wild-type counterparts. So, I'd just like to summarise the take-home message from my talk. We've identified that DNA repair factors are important determinants of seed figure and viability. We've shown that ATM, which is a master controller of the plant cell's response to DNA damage, controls progression through germination, that is, it regulates the lag period that we see in low vigor seeds, and both ATM and ATR are regulating seed viability. And we propose that ATM and the DNA damage response are safeguarding the plant genome in germination from high levels of DNA damage experienced during desiccation and quiescence. And finally, our most recent work has established that DNA damage signaling through the SOG1 transcription factor is resulting in programmed cell death in aged seeds, and we hypothesize that it's acting as a quality control mechanism as seeds deteriorate to eliminate damaged cells. And finally, I'd just like to thank the group leader, Chris West, and the support of other members of the West group. And I'd particularly like to thank Bill Fringe-Savage and Steve Futtit from the University of Warwick in the UK for their collaboration on the ATM work. And I'd like to acknowledge and thank the financial contribution of the UK Research Councils and the EU EcoSeed project. Okay.
Thank you, Wanda, for a very interesting, illuminating presentation.